So just a brief thing that I saw that really made me think about what's happening, and thought it made me think of all of you as well, is first, make sure you take some mental breaks as often as possible. Instead of reacting to every thought, try to let it go, because your reaction is what keeps negative thoughts alive. Because I'm sure some of you are thinking negative thoughts. The key thing is to try to think positive. I know for some of us, it's definitely not a positive at all, but there's always a positive lining for everything. Like for me, I've been pulling my hair out, trying to figure out what we're doing for online in terms of positioning stuff, but then I learned about Kahoot and I learned about Zoom, and so there's a positive with that. Also stressed out about my 95-year-old dad that's sitting at his house, and every time I bring him food, I always wonder, I hope I don't get him sick for that. So it's definitely stressful, and I definitely understand that. I haven't seen my dad in 3 weeks now, because I started to do this before it actually became, when I started hearing about it, I started to do that because I was a little concerned about it. One thing I may do is for extra credit, I may put an assignment on the coronavirus, because I think it's really important to kind of understand a little bit about it, because sometimes fear of the unknown sometimes makes it stressful. So having an assignment, having you look up this for extra credit may help alleviate it a little bit, at least as much as it can. So again, so be conscientious and active in your mental space so you don't get overwhelmed, because I'm sure that's what some of you thought as soon as you heard it was online. The first thing that was flooding in there was a whole bunch of negative thoughts about not being able to do it, which makes you feel overwhelmed. And so you've got to switch that off. Remember the beginning of class when everyone was super stressed out about, oh my God, there was a lot of stuff, and a lot of you came around. It's a routine, the same type of thing. It's going to be the same thing with this. So why don't we go ahead and get started and seeing what we're going to be in for. Good. Okay, come on, click. There we go. So everything that I'm going to say here is also on Canvas, so you don't have to freak out and try to remember everything that's here. All of it's been organized. I have a message to students that is posted at the very, very, very beginning, so you will know what's happening. One way that I did organize a class, I have taught some online classes, so I tried to organize it like that a little bit to try to make it a little less crazy. So each week is in a module, and I have provided each week with an announcement that has what is due for that week. Right, the best I can. So if you see typos or things like that, I really apologize for that, but it's been crazy trying to type those up. So those are in there. I will also send weekly announcements reminding students what is due each week. I've been doing that, but it's definitely going to be key for now. Also, any resource material that includes the review sheets, the videos that are there, the frequently asked questions are all in the same modules that we had before. The only thing that's different is now I kind of rearrange stuff and put some of these categories in the weekly ones to make it easier for you to find. But just know that the review sheets are still there, they're just in the modules that they're originally in. Any light blue links, not the dark blue links, but the light blue links are either documents, so that's what you click on to get the document, or it will take you to a new site. So the beauty of Zoom is, I love this, so I can actually take you, and you can see this then. All right, so let's go ahead, and this is what it kind of looks like. This is my view, so it's going to be slightly different for you. So let's go ahead and go in here for, just to show you. So this is the message that has everything. I love Michael Bublé, so I had to put him in here. Anyway, so if you go down here and you see these light blue links right here, these are things that either have documents or they take you someplace else. So if you don't know where a Word document is, it's going to be in a light blue link that looks something like that, if you haven't been online yet. All right, and I'll be back on here because I do have some things I want to show you anyway, with where things are. Okay, that's not the one I want. Where is it at? All right, so each week there will be an announcement page. It's already set up, and if anything changes, I will let you know. All the written assessments now, so that means the chapter notes, the extra credit worksheets, anything that was like like that. That was something that I printed out and gave to you. Now they are Word documents. And so all you have to do is type them up if you prefer writing them, and you can submit and you can make it a PDF as one PDF, all right with that as well. Just be aware that they're not set up for PDFs yet. And so there's no longer a way to email me the submissions. You have to submit through the actual Canvas site. So why don't I go ahead and show you what that looks like. You have not done this already. All right, so if you go to our home here, and let me go to student view so you have an idea of what this looks like.
Então, vai.
Settings. Let me go to view. All right, let's take a look and see what one of these look like. Which one has one? All right, let's go. All right, so does everyone see this big button up here that says submit assignment? So that is where you're going to actually post your assignment. This is where the assignment is. So you're going to click on that and it's going to become a Word document. Save that Word document on your desktop, fill it out, and then you're going to hit submit. All you have to do is click File Chosen, and then it will take you, just add your file, open, and then you submit. Any questions on that? So that's what you have to do for all the, so this one's already set up for PDF. So it will tell you up here whether or not it is or not. So it'll accept doc documents, docx documents, and PDF documents. So any questions on that? All right, let's go ahead and you can come back to this later if you need to look at it. All right, so again, another thing too is if you do not have a reliable Word or PowerPoint using software, or if you don't have reliable internet and a computer, please let me know. It looks like I have a chat. So great question. So Emily, cannot remember which section you're in. So if you're on Tuesday, Thursday, we are going to start up on Tuesday with Chapter 8. If you are on Monday, Wednesday, Friday, we're done with Chapter 8. So what we're going to do is, looks like you. I didn't really think about what we were going to do because we're a little ahead on Monday, Wednesday, Friday. So that is what we're doing there. So hopefully that helps. Right now anyway. So it looks like Chapter 8 or a little bit of review. So if you're on Monday, Wednesday, Friday, we probably won't have a long lecture that day. Maybe some review problems or something. No, so great. Thank you for chatting. So you can chat. So make sure if you do have any questions, you can always use the chat box. Do not know what that is. Okay. Oh, yep. So something to remind me to tell you, in your modules, in the week 8 module, week 9, I think. Week 9 module, the first one that's up there, please take the online survey as soon as possible, just so I know anybody who is at risk of not having certain resources available to complete the class. So that one's really important. All right, so let's go ahead and go through each part and we'll discuss how it's going to be handled. Lecture, you're seeing how it's going to be handled. So it's going to be just like this. And it'll be, what's even better, because now it'll be recorded and you can listen to it later if you can't make it during that time. So you definitely want to make sure that you tune in. So right now it's scheduled for every time we have a regular lecture hour, that is when we'll have class. That might change depending on, oh, was someone drawing on there? That's not me, by the way. Times might change depending on how much attendance we get. If I see that we're not getting half the class coming, it may switch to, that particular section might get switched to once a week. And then that is the lecture that you'll get for that week. So we won't just have a live one. But it will be through Zoom. As soon as I get the account set up in Anne Arundel, I don't know why it's not working, but as soon as that's set up, it'll be a scheduled meeting that's in there. It'll be easier to click in instead of me just sending emails to everybody. But until that point, I will be sending you emails when they will start. The beauty of this as well, or a little bit of the downside to this for the recordings, is that once it's done recording, I am going to post it on YouTube so I can get the transcribed version of this. So the posted video on Canvas will not be available for at least one day to allow for the transcription piece of that. So just be aware of that. There is going to be a little bit of a delay there because I have to physically get that done. All right, in terms of attendance, there is no formal attendance anymore because of how this is set up. And I know some students can't make it at the designated time, but we still have to record attendance for financial aid. So what will happen is it's really going to be based on your engagement in the class. So what gets your attendance part is to attend the live lectures. So then you get your attendance for that. I know that some students can't do that, but there's some other options if you can't make it for a particular lecture, for example. Just make sure you complete all your lectures on time or your stuff on time. That's really the main thing. So we have everything set up for either Monday, Wednesday, Friday, or Tuesday, Thursday, because I wanted to keep it the same as we've been doing it. But in some cases, there'll be a little bit of leeway because I know there's a lot going on. Another way to get your attendance points is, or not points really, but your attendance is making sure you take your quizzes and your exams at the designated times. What are some of these things here? So Mentimeter no longer will be graded, and that's because I can't ensure that everyone's going to be there. So I need to go back and make sure everyone's gotten their 9 sessions in there, done all the sections yet or not. So that grade is done. All right, so that is what you'll get. You'll be graded on the 6 that I have on there, and 3 of them will be dropped. But we'll still do it in class to make sure that I'm gonna still ask you questions. That's not gonna change, it's just not gonna be graded. All right, some things that did change. So there's a question for me, let me take a look. All right, so do we still have April 7th to complete all assignments till then? So that's a great question. So it depends on the due date of the assignment. So some of them I haven't changed yet. You can start completing them now. So it's definitely not, if you have the opportunity, you can complete some of the assignments early. So what will happen is it depends on the assignment. Some of them I will go through and let you know which ones there's no extended due dates for. For example, Connect, which I'll talk about in a second. Unfortunately, that's the one that is stuck. So if you don't make up that time, then it won't be counted. Everything else will be a little bit, I'll let you know which ones are not. So everything, I have not changed all the dates yet because I cannot remember which section you're in. So it depends on your section. I haven't, Monday, Wednesday, Friday, I have not changed the dates yet for those. For section 10, I haven't changed the times yet. So those are still not done yet, but those will be worked on next. All right. Any other great questions? Any other questions? All right, so some key changes. Something I'll talk about at the end is the final exam percentage is lower. And so 10 percentage points was taking off, and so they've been redistributed to other sections. So the first one is now Connect is worth 15% versus 10%, which means that this is your opportunity to start plugging away at those Connect assignments because that's easy 15% there. So those have not changed. It's going to be the same. They're going to be due on the dates listed. The syllabus has been updated. The schedule has been updated and posted already. So those are already up there in Tuesday, Thursday sections, but I'll get to everyone. Sorry, Monday, Wednesday, Friday, I'll get to you as soon as possible. But those are not going to change. For chapter notes and outlines, this is now, instead of working 3%, now it's worth 8%. So again, this should be really, really easy. You just do it with your Connect readings. fill them out as you're doing them. I'm grading it on completeness, not correctness here. So you want to make sure that you fill in all the blanks. Don't lose points because you didn't fill in something. Try the best that you can. Use your lecture notes if you have to, because that's where some of the answers are. You can complete these again using Word document or posting as a PDF. This is one of the assignments where you do have some leeway with. So for example, say it's due on Tuesday, and for some reason you can't do it on that Tuesday, I can give you some leeway on that. You just have to contact me first and let me know, all right? Because it will be still scheduled for that time, but just let me know. And then most things, all things have to be turned in by Saturday if there is leeway associated with them. All right, so for example, if it's Tuesday and you can't make it, then you have until that Saturday to make sure you get it in there. After that point, it's done. There's no doing that. The other key thing is make sure you contact me. Because remember, this is part of your attendance. So if someone continuously turns them in late, that will reflect on your attendance. It's okay for once in a while because things do happen. But if it's constantly happening and you don't tell me about it, then that's going to start reflecting on your attendance. And so any questions on these two? All right. There we go. All right, so the in-class quizzes. So these are going to be slightly different. They are still 2 points, so low in terms of amount. They're going to be posted on Canvas now. So slightly different format here. So what will happen is it'll be in the same format as the homeworks. And so based on, and again, I have to emphasize this is kind of a group decision as a Biology 101 group. We made these decisions in terms of timing and things like that. So many of these questions will be considered a half a minute to complete. So for this case, there's 4 questions. I will give you 3 minutes. All right. This one, students will have a 24-hour window to take that quiz. So for example, if you have Monday, Wednesday, Friday, and you have 9 o'clock class, you will have 24 hours from that class to turn in that quiz. All right, you have 24 hours. All right, so some key things about these. I should say there are 4 multiple choice questions for these, so they're all multiple choice. There's no fill in the blank. There's nothing like that. And I have a demo because I wanted to show you this because I know some of you are stressed out about this. So let's go back to the, oh, I need to do something else first. Let me go back and go into, oh, close out of here. Never mind, let me see. What was I at? What happens when you have a gazillion windows? All right, where is this class at? Oh, here it is. All right. Let me log out here and let me start the quiz. So please don't do this quiz. This is just a practice quiz to show you what it looks like. But I wanted to give everyone a chance to see this because I know I have some students that are stressed out about this. All right, now I'll go in here as a student. All right, so this is when you take a quiz. So you're going to see the information. Please read the directions. All right, so this is important. For these quizzes and exams, you will not be able to backtrack. So you have to answer the question as is. Also, is there anything else? 1 question at a time. So we'll take this quiz to give you an idea. So this quiz is just a minute. All right, it has 4 questions in it. It's just to give you an idea. So if you're ready to go, whenever you're ready to do this, if you push the button, you have to take the quiz. All right, so you push your button, right? So once you submit an answer, you will not be able to change it. So it tells you that. All right, so question 1, the microbe that's currently in the news for causing the pandemic infection is what? Anyone know the answer to that?
Coronavirus.
Coronavirus.
There you go. So it's corona, right? So you pick that. Next. Notice I did not hit submit because if you submit, then I will take your answer. The microbe in the news. Where?
Microphone means microlabs. Like, I'm just joking. It's definitely China.
You sure? No, it's not. All right, so it's China. All right, so next. How does the microbe spread to other people?
Drinking a Corona. That sounds
Anybody?
Arousal and contact, close contact.
Right, so close contact, right? Okay, so next. Where does cellular respiration occur in eukaryotic cells? Oh, so we ran out of time. That's okay. So what was that answer? Mitochondria, right? Oh, yay. Okay. All right. So that gives you an idea of a minute of time. So we went through how many questions? About 3, and we talked about it for a few minutes. It really, again, you just need to take your time and read the questions. The questions are not going to be super long for this. All right, so they're not going to be long. So I know some of you are really stressed out about this. Just take a breath and look at the question. You will know these. These are things that you've seen before. All right, so any questions on how to do this? As you saw, once it will just submit it for you once the time runs out. So you don't even have to submit it.
I don't have a question about this exactly, but will we be able to get an updated list of when items are due?
Yeah, so they're already, are you in Monday, Wednesday, Friday section?
Tuesday, Thursday.
Okay, so if you go to your section, let's go, this is a Tuesday, Thursday section. Come over here and you'll see stuff over here. This is the modified schedule here. This is a message that's going to have the same things I'm telling you right now. And if you go to these weekly announcements, each module will have them. So you will scroll down, it will tell you exactly what is due on each day and how it needs to be submitted.
Thank you.
No, not a problem. Anybody else? I guess. Yes, on the Zoom, this will be, the Zoom meetings will be recorded and I will post them. All right, how many subs do you have on YouTube? I'm not quite sure what that is. I am not very good with the tech savvy stuff. So you mean, if you mean any other videos, I don't yet. So right now they're all private right now. Right now I'm just trying to make sure that we have some of these lectures posted. I don't know what I'm gonna do with them in the future. That might be something. I'm gonna have I made a YouTube channel up just to do this. So more on that to come if I decide to do that. No, so great questions. Go ahead back here. If you have any other one, just go ahead and feel free to chat there. Oh, I got another one. Here we go. Nope, I thought there was another one. I don't see it there. All right. In-class activities. All right, so this one, again, a little bit more challenging, but I came up with a solution. So some of the students have been telling me that they like Kahoot, so I've been looking at that. And so that is how we're going to do our in-class activity. The reason why I chose it is because students that are not in class can actually participate in this activity. So that's why it'll give you a chance to do this, except you have to finish it within the time window designated yet, and I just don't know how long that time period is going to be. But I do have a demo, so everyone can get excited about this, because I thought this was really cool. All right, let me see. So what you'll need for this is you'll need a phone or something. You can even, I think you can even do a separate window to do this. So how Kahoot will work is something like this. All right, so we're going to play classic here. So it looks familiar, right? This is almost exactly like, and I will turn on that music because the music is kind of annoying for right now. All right, so hopefully everyone can hear me. I hope I didn't turn my sound down too much. So go ahead and join at that web address right there and put the pin in.
Wait, we don't have to do this right now, do we?
Nope, you can watch. It's just to get us in practice. I wish I could just turn this off, really. Let me see. Oh, there it is. Yay, okay, now I can turn my sound up. So you don't have to do this right now, but this is just a demo to get everyone understanding how this is going to work when we do it in class. You will also, if you can't make it to class, you will get a pin or a link to be able to do the Kahoot that's associated with the class. I will say this for the Kahoot that's going to be graded. Make sure you put your name, your first name and last name to get a grade. You can do the fun one for this one, but the other one I'm doing it for a grade, so you want to make sure you put your name. So preferably your first name and your last initial if it doesn't fit in there. Have fun with it now as long as it's not intrusive to anyone or offensive. Okay, do we have any more students that want to do this? Let's see, I have a chat. See if I can get 20 people in here and then we'll go ahead and get started so you can actually see what this looks like. All right, so the name of this game is it's going to be multiple choice questions or other kinds of questions, and you're going to be racing against other students to be the first to answer these questions to earn points. So this is actually a game. All right, there we go. Anybody else? Last call. If not, you'll get to try it when we actually do the All right, so why don't we go ahead and get started so we can see what this looks like. So I'm going to put the start button. You're going to have 20 seconds to answer. There's like 3 questions because I just wanted to do a quick demo. All right, so here we go. Everyone else, you can go ahead and watch and see what this looks like. You should be able to see it from my share screen. All right, so we got 23. All right, let's see how it goes. All right, now pick the correct answer here. When they pop up, you'll see different colors. Pick the one correct. And you're racing against everybody. All right, so we got 1 more second. There we go. All right, so the answer is enzymes. All right, so let's see. Oh, let me see. So notice that everyone gets points for that. So the quicker you get in, the more points you earn. All right, everyone ready for this is a puzzle one. Now you need to put what is the order of steps in cellular respiration. Put them in order. You can slide them. Okay. Whoops. Sorry about that.
Leah.
Sorry about that. I didn't mean to click on it so you can see the answers. So that was glycolysis, pyruvate oxidation, Krebs cycle or T-scape cycle, and oxidative phosphorylation. right, so if you see a triangle, I want to say that that means that you got them, they're doing the bonus thing, I think, with that. You get bonus points if you do it quicker. All right, so we have one more question. Everyone ready? The last one.
Oh, he's big.
All right, so the answer is hypertonic. So when a solution has more solute, that means it's hyper, like hyperactive. All right, so here we go. There we go. Let's see who number one is. All right. All right, so no, we can stop sharing that now. So any questions on that? Okay, that did not stop it from doing that. Okay, let me go back. All right. Any questions on that? That will be the review. So what will happen is, so what will happen is if you're absent that day, there's going to be a challenge one, which will be the same thing, but you'll be against yourself. It's just earning points. You just have to do it within a certain time. I'm not sure if I'm going to make it 24 hours or 48 hours. I haven't really thought about it yet. But that's how we're going to do that. So any questions on that? That's All right, so question. So the Kahoot, this was not the Kahoot app. That was actually, it was on mine, so I can invite you to do it. So you just use your phone. You just you just type in the link that's at the top, and then you type in the pin, and you don't even have the app. You don't need the app for that. All right, so great questions. All right, chapter homework, exactly the same. That hasn't changed at all. So that will be less stress because it's the same. All right, so the exams. I know this is the one that is probably stressing everybody out. So let's go through this a little bit. So the first two exams, so 2 and 3, the point values have not changed. They will be located in the weekly modules when they're complete. These will be quizzes just like the quizzes I demonstrated to you. So they will be posted there. They'll be available for 48 hours from the start of the class. So let's just say that you are my 8 in the morning class on Tuesday. You will have 48 hours from that time to take it. Students will have one attempt to take it. So please make sure you have enough time to take it. You also want to make sure you have a reliable internet and a reliable computer while you're doing that. You cannot log out. So it's going to be for, in this case, 25 minutes. So I do apologize that it is greatly condensed. However, all the questions are multiple choice, and they're going to be a little less wordier because you have to get through the questions in that sense. So just be aware that it's going to be about 30 seconds a question. Again, this is the consensus that was determined by we actually did this as a team. So that was the number that we came up with. The exams are actually going to be more of a pool of questions. So keep that in mind as well. So this is going to be a pool of questions on particular topics. So you want to make sure that you are ready to go and take these exams. So these are not open book. So when you are ready to go, you need to be ready to take it. After the exams are done. Yes.
So for the exam, will it be a lockdown browser for them, or is it just going to be through Canvas?
It's going to be through Canvas.
Okay.
It's part of the reason why the time is shortened for that reason. Let me see.
And if you do need like extra time for
I'm
that,
sorry?
you could just email. You said if you did need extra time, email you.
Sorry, you're breaking up.
You said if you need extra time to email you.
Yeah, I'm sorry. I am. So go ahead and type that in for me because I'm missing every other word. So I apologize for that. Yeah. So part of if the question is, so part of the reason why the time is being condensed as much is because we don't have the lockdown browser option. So that's a good question. So for the extra time piece, unfortunately, that is set for students for taking the exam. So I will not be able to do that. So until we get word otherwise, I'm going to have to keep it at that time. So if we're actually having another meeting this week, so I'll see if anything changes with that. But currently, that is what's going to happen with the exam in terms of time. If you're interested in any of the exam content afterwards, please let me know so I can discuss that. Let's see. No, unfortunately not. So this is a faculty only meeting. All right. So in terms of makeups and things like that, so this particular, it's going to be the same. So if you cannot make in this case, you have 48 hours to take the exam. Again, this is put into place so students have more of an option to say that your work schedules change and you can't make the regular class time. At least this will make up for the inability to be in class. So there's a more of a range that you can take the exam. So you do have 48 hours to take that. If you can't make it within that 48-hour window, you need to contact me. All right, so we need to talk about a plan on doing that, how it's going to occur and stuff like that. So please contact me if that happens. All right, so exam, the final exam. So the final exam, slightly different in different ways. Number one, it's no longer 20% of the grade, it's only 10%. So that should be a good thing, right? That's a positive thing. It will be put into two parts. So there is an exam 4 and a cumulative piece. So there's actually two parts here. So that's also a good thing too, because now you don't have to take them all at one time. All right, in terms of point values, I don't know all the point values yet. So that is something I will let you know about. So this will be located in the week 15 module when that's complete. Same format, it's going to be a Canvas quiz. All right, so exam 4 is going to be the new material piece. We have cut out the evolution piece, so it will focus just on chapter 14 and 15 for that. Let me see what my question is here. All right, so that test will be 100 points and it'll cover those two chapters. And then the cumulative piece, I will give you more information on that. I don't know how much that is going to be worth yet. So I will let you know as soon as I know that piece. Let me see. Students will have 48 hours to complete that as well. So what I would do is I would pick one of the two sections, do that first. I don't know which one you would prefer to do. It would be really what your strengths are. So if you're really good with exam 4, then I would do that one and then spend some more time on the other piece. But you want to make sure that you get those two pieces done. Same thing, you'll have one attempt to do that. So you want to make sure that you sit there with that. Again, I don't know about times or anything with this one yet. Again, these are not open book. And also, same thing with makeups, a little bit different for the final exam because classes have to be done by the 22nd. That is our deadline here. right now, if I remember correctly, so if you're interested, so Tuesday, Thursday, your exam is on May 19th. That is the final day. And then for Monday, Wednesday, Friday, yours is May 20th for that. So extra credits. So extra credit, you will still have the opportunity to do worksheets. They are now posted on Canvas in each of the weekly modules. So same thing like the notes, just make sure you complete it, type it up, or write it if you can do that, and then you make it into a Word document or a PDF for that. Additional extra credit, I will be tutoring. I think I have that up on the next slide. So we will be having science tutoring hours on Zoom. I will email the SI one, so everyone should get that email when my meeting starts, so you can get that link. And I'll be on Zoom Monday, starting on April 6th from 10 to 12. So I'll be online, so you can ask me live questions, or Thursdays, starting April 9th from 1 to 3. This is your way that you can get those extra credit points for attending tutoring if you didn't get a chance to do that. Let's see. All right, so in terms of other things, so I do have a couple of discussion boards up, and these are not for grades. These are just for asking questions. So say you have a question, you'd like other students to help you out, or myself, then you can, there's a questions one, and then there's also a concern one, because I know some students are feeling concerned about either the class or whether it's the virus or whatever it happens to be. Using that discussion board, you can talk with that. I should be getting pings on when students write me on that, so I apologize if I don't get on there right away. I will get on there as soon as I get a ping. I'll try to remind myself to go look there as well. You can always email me as well, if personally on that, if you don't want to use that. Using the discussion boards, please make sure, again, use your online etiquette. So you want to make sure it's a safe environment for everyone to express themselves, and so please be respectful when using those. All right, so any questions for me right now before we start the review piece? I know we went through a lot. So this is taped, so you can look at it later. All right, so great. So that is a very good question. So labs. So what will happen with labs is labs will be online. And what will happen is there's going to be videos, there's going to be photos, and these are going to relate to the lab. And so what will happen is they're going to be set up in, I want to say modules. Students will have to look at the module work, or stuff that's in there, answer questions that are in the lab manual, and then you will have to take a quiz at the end, not the end of the lab, but you will take a quiz sometime that's going to be about that lab. And I want to say it's 10 questions, and it's multiple choice at this time. Your instructor will be handling the grading and things like that. So let's see. A great question on the exam 2. Exam 2 is on the schedule, so that's a great place to start looking, but just to kind of give everyone a heads up, exam 2 for Monday, Wednesday, Friday is on April 8th. For Tuesday, Thursday, it's the 9th. That's when they'll be opened. All right, so that's when they'll be open to take. All right, so right now, that is what we have. I think that's part of the reason why we don't have all the answers for the lab. I tried to give you a little bit of kind of like what's going on, because I know people are kind of stressed out about what the heck's going on. We start on Monday and we have no idea. But that is what's happening right now. So it's going to be online. It's going to be based on the manual, except for 2 labs. There's actually going to be 2 labs. They're going to be virtual labs that you're going to use the Connect for. So just be aware of that. There will be 2 of those. I want to say it's the DNA electrophoresis lab, and what's the other one? There's going to be blood typing, I think, is another one. Let me see what I have for a question here. All right, so great question. So there's going to be videos of the lab being conducted. I think there's going to be videos of the specimens and things like that. And then you're going to be answering the lab manual. I'm not sure if there's going to be quiz questions as well. I haven't seen that yet. So I don't think those are graded. You're going to be graded on your quiz. But I'm not sure if there's going to be accompanying questions that are going to be with the actual video, or they're just going to coincide with what your lab is. I want to say that it's going to go in line with your lab manual. So for example, when we did the plant lab, and you had to look at the different, let me see, like the tubes. So for the tubes that you put underneath the light, so it would be a picture of the results, maybe a video of how it was done, and then you would need to answer the questions that would be in the manual. So I want to say that's how it's happening. Of the day of the lab, right now what's been suggested is that instructors meet with their students at the time that the lab meets. So if your lab is at 2 o'clock on Thursday, then you meet at 2 o'clock on Thursday, and the instructor should give you some lab instruction. So introduction to the lab. If you have me for lab, you know I have the PowerPoints and we have the stuff on there. So each instructor should be contributing some kind of thing like that. So the quiz time, that I don't know yet. I don't know if it's going to be due, there's certain, you know, if it's 24 hours or if there's a time restriction on that. I haven't gotten that far yet, but that hopefully that gives you more of an idea what's happening with lab. All right, so any questions? Again, also you can always email me if you have questions on lab as well. I can't give you answers to the questions per se, but I can help you with concepts and things on that if you get stuck on some of the lab stuff. All right, so any other questions on the lab piece? All right, so everyone ready here? So so in terms of giving us some things to study for the exam, because it's a pool of questions, it's going to be slightly different. I'm going to go over some more concept types of things, some things that I would like you to focus on. So it's going to be a range of questions for these, but we'll do the best we can with this and focus in on some of these things. So in terms of the topics, it's going to cover chapters 5, 6, 7, and 8. Again, it's going to have 50 multiple choice questions. They're going to be a little bit shorter, so they're more doable in terms of answering it. So it shouldn't take you as long to read the questions. Some guides in terms of what to review. So I definitely would review the homework questions, the quiz questions, the past quiz questions, the extra credit worksheets, the Mentimeter questions that we had in class are really a good guide. Also, Quizlet will help a lot with this. So it's a good time to use electronics to help look at the vocab, look at the questions. Also, something else I forgot to mention is that at least for Lab 10, I can't guarantee it for other labs. What I've done is I've created a notes that will go with the lecture for Lab 10. So you can use that as a guide. There's no points associated with it, and you don't have to use that to take notes, but it's found in the module. So if you want to use that and type up your answers while we're going through it, or write in your answers if you want to do that while we're going through it or for later, just please feel free to use that. I just can't guarantee if I can do the other ones because of time. So I still have to work on notes for chapter 13, 14, and 15, which I have not done yet because of all this. So definitely, if I get a chance to, I definitely will do that. And I think that will help focus the lecture piece a little bit. Not moving. There we go. All right, so let's do each chapter like we did for the last review. And so hopefully I'm going to test you a little bit, see where you're at with some of this material. Pictured on the left, that is a picture of a membrane. So you need to remember all the parts of the membrane that we went through. So we talked about the phospholipids, we talked about cholesterol, we talked about glycolipids, glycoproteins, we talked about membrane proteins, the 2 different kinds. So you want to make sure that you know those, because again, those are some definition type questions. That could be a definition. So for example, on the bottom, that's a picture of a phospholipid. We definitely had some questions on which part of the phospholipid is has a negative charge and interacts with water, and then which part is the part that interacts in the middle of the membrane. So you should know those. So make sure you know the polar head and the nonpolar tails. All right, so you want to make sure that you remember those. All right. So in terms, so I'll ask this question. You can put this in the chat box if you want. So for things, what kinds of things can go through the membrane rather easily? What are some of those things that can go through the membrane rather easily? Other answers? So Kurt offered an answer. Any other answers? It's been a while, right? So this one's been definitely a while. All right, so things that can cross the membrane easily include our non-charged molecules, so things such as gases, so oxygen gas, CO2 gas. Water, for the most part, can go through, although it usually uses porins to help out, but it can also go through. Hydrocarbons can also go through. So those are molecules that are made of carbon and hydrogen. So those also can go through easily. Pretty much everything else we've talked about in class does not. So the macromolecules, so that's your proteins, carbs, not lipids, proteins, carbs, nucleic acids, those don't go through the membrane. And then ions. So ions also don't go through the membrane. Don't forget large and in charge. So those are the ones that do not cross the membrane and stay on their side. The only way that they can come through is if they have a protein, right? So a protein is like a doorway, so it helps them to come through. All right, so then we talked about passive and active transport. Passive and active transport. So this, again, there's a lot of questions that can come in this category from what are some examples of passive transport to what are some examples of active transport. So you definitely need to know the 3 types of active transport that we talked about. You need to know about passive transport in general. So does anyone know passive transport? Do you need energy or not for that type of transport? Do you need energy for passive transport? All right, so no, right? So you don't need, I'll try to put this over here someplace. So you don't need any. So active transport you do, so you definitely need to know those. So which type of transport do solutes or substances move from a low concentration to a high concentration? So which one is that, active or passive? Which one is that? All right, good. So that is active. So again, that's another piece of information that you definitely need to know. Some other things we went over as well. We went over equilibrium. So you definitely need to know what equilibrium is. So equilibrium is just the movement of particles until they are equal on both sides of a membrane, for example, or just spread throughout a area. In the bottom here, so we move the Zoom chat so you can see it. So for the bottom, those are some different types of active transport that require a membrane. So we talked about phagocytosis, which is where you take up large things. We talked about penocytosis, which is liquid drinking. So we talked about that. And then we talked about receptor-mediated endocytosis, where you have things bind to it and it triggers stuff to be taken in. So we talked about that as well. All right, so I do have some example problems for us because you're gonna have a problem like this on the exam somehow. So you wanna make sure, again, you wanna have a little bit of time dedicated to this kind of question. So this one will require a little bit of, you actually have to read the question. for this. So let's go through these. So starting with the one on the left, the beaker. The beaker is what kind of tonicity as compared to the sausage? Anyone remember? So what is that? So the beaker as compared to the sausage. So good. Very good. So that means that the sausage has what kind of solution as compared to the beaker? Good, so that's also isotonic as well, right? So it's going to be isotonic. All right, so which way is the water going to move here? How is the water going to move? Good, so it's going to move equally in and out, right? So it's going to move equally in and out. So what's going to happen to the sausage? Is anything going to happen to the sausage in terms of size? Yeah, nope, so it does it the same, right? So it stays the same here. All right, so let's move on to the next one. All right, so beaker. So the beaker solution here is what has what kind of solution compared to the sausage here? Good, so that is hyper, right? So that's hypertonic, which means that the sausage has what kind of solution in it? Good, so it is hypo. All right, so how is the water going to move here? How's the water going to move here? Good, so it's going to move out of the sausage, All right, so what about the sausage? What's going to happen to the sausage? Good, so it's going to shrink, right? So it's going to shrink. For right now. All right, so let's take a look over here. So beaker solution, what are we going to have here? So beaker solution is hypotonic, right? So this one's hypotonic. Okay, there we go. All right, so that means the sausage will have what kind of solution then? Good, so it's going to be hypertonic. So which way is the water going to move now? Good, so it's going to go inside. And what's going to happen to the poor sausage? All right, so it could burst, right? So it could burst, or it's just going to get bigger. Depends on your knot, right? If you tied a really good knot, it would be all right. All right, so any questions on how to approach one of these problems? So the key thing is to make sure you read whether it says beaker, or it says sausage, or it says cell, if it says plant cell, if it says animal cell. Really make sure that you tune into what the question is saying. Usually I will try to underline those particular words to make sure that you know that that's where the focus is. All right, so I'm going to keep going, and then you can always chat in there if you have any questions. All right, so we also talked a little bit about some active transport methods. So here's some here. So we talked about aquaporins. We also talked about sodium potassium pump. So some key things, the key takeaways to that sodium potassium pump, because if you plan on taking any other science or any other anatomy physiology or other classes like that, you're going to see this again. I know a lot of you are not, but just in case you are, you're going to need to know this. So what are some key things to take away here? Number 1, sodium ions leave the cell. All right, so they leave the cell. There you go. 2, potassium ions, they come into the cell. So that is the direction of the diagram. So when you're looking at it, these sodiums are coming from the bottom. Right, so they're coming from the bottom in. I don't know why this diagram is backward. So it comes in and then goes out. All right, so ATP is needed for this type of transport. So what is this type of transport then if you need ATP for it? Okay, so this is an example of active transport because you have ATPs that are used here. And so ATP here, how it provides energy is adding that phosphate group on there, right? So it phosphorylates the enzyme. All right, so does anyone remember how many sodiums leave the cell? How many sodiums leave the cell? Good, so it's 3 out, and then how many potassiums come in? That would be 2, right? So 3 sodiums leave, 2 potassiums come in. All right, so any questions on that? All right, so you're doing awesome. All right, so here I have some animal and plant cells, so definitely you'll have to be familiar with these. All right, so over here, we have 3 different blood samples here. And I'm not going to give you a picture, but these are just, it could be a description that you may need to know what's happening here. So a red blood cell, if you put it into distilled water, what do you think is going to happen to the red blood cell? So you put the red blood cell into just pure water, remembering that pure water has no solutes in it. Okay, so good. So it's going to burst here, right? So you're going to have hemolysis here. Red blood cells are animal cells. They don't have a cell wall, so they're going to burst open. All right, so down at the bottom, so 0.9% saline. Any guesses as to what's going to happen there? I'll give you another hint. The other solution is 2% saline. So what kind of solution do you think 0.9% saline is to the red blood cell? And we did this in lab too. Any guesses on that one? All right, so let me give you another hint. The inside of the red blood cell also has 0.9% saline as well. So that means that the red blood cell is in a good, so isotonic solution here. Isotonic solution. All right, so that's what red blood cells normally look like. All right, so maybe this will help out then. So if you put the red blood cells into 2% saline, what's going to happen then to the red blood cell? If the inside of the red blood cell is 0.9% saline and you put it into a 2% saline solution, what's going to happen to the red blood cell? Anyone remember? Good, so I see the term. So what is shrink when we talk about red blood cells? Anyone remember that word? It starts with a C. Anyone remember? All right, so we're talking about animal cells. That is crenate. So crenate. Again, make sure that you're familiar with those terminologies when we talk about animal cells, and then it's different for plant cells, right? So plant cells is different. All right, so over here, anyone know what this is here? So this is an example of what? A plant cell that is undergoing what? Anyone remember? So the cytoplasm is pulling away from the cell wall. So that one is, it looks like I found the slide that everyone needs to review a little bit. All right. So when you put a plant cell in a hypertonic solution, its cytoplasm is going to pull away, and that is plasmolysis. All right, so the next one. All right, so anyone know what that one is? I'll give you a hint. That one is isotonic because the arrows are going in and out the same way. Can everyone not hear me anymore? Is everyone good? Hello? Can anyone hear me? Okay, I just want to make sure. Okay, so the second one is isotonic. So when a plant cell, oh good, there it is. All right, so that is flaccid, right? So flaccid's a term when a plant cell is in an isotonic solution. All right, so the last one, what's the last one then? Anyone remember? Starts with a T. Anyone remember that one? There you go. So turgid, right? So if you put it in a hypotonic solution, it is turgid. All right, so please make sure that you review this. All right, any questions for me on this part? All right, let's continue. So that's the end of chapter 5. Let's move on to chapter 6, which has to do with metabolism. And I think I told everyone here, this is one of my favorite diagrams because it kind of ties everything together with what this chapter is about. So you want to make sure that if you can look at this diagram and kind of have a basic idea what's happening, that will help you with starting to put together the detail piece of this. All right, so don't forget that just chapters 6 and 7 really just deal with how a cell makes energy to run things. That's all those 2 chapters were about. And then plant cells, or the ones for photosynthesis, is just how do we get the glucose to run everything in chapter 7. So that's what that chapter is about. All right, so some topics to review. So the laws of thermodynamics, make sure you know those. That one's not too bad. Questions for me? No, not a problem. Thank you for coming. All right, so laws of thermodynamics. So the next one on the left, again, you could have a diagram. These are 2 types of reactions based on energy. So does anyone know what kind of reaction is on the top? So someone asked me about the exam. So it depends on what day. It's either April 8th or April 9th, depending if you have a Monday, Wednesday class or a Tuesday, Thursday class. All right, anyone remember what the one is on top? So I guess I'll give a hint on that. So that one is, oh, there we go. Whoops. The top one is exergonic. So the bottom one must be then endergonic, right? So you want to make sure you take a look at those. All right, you need to know the definition of those. So these are just diagrams of those. Remember, exergonic are reactions that release energy because the products have less energy than the reactants. Endergonic reactions are where the products have more, so that means you have to put more into it. All right. Anyone remember which one is spontaneous? Anyone remember which one is spontaneous? All right, so good. So is the exergonic one, because remember it's releasing energy. Releasing energy. All right. I have an example. So here we go. So we have an equation. That equation hopefully looks familiar to everybody. Its delta G is negative 686 kilocalories per mole. So is that inorganic or exergonic? And another one you need to be thinking about, is it spontaneous or not? So what's the first one? So is it endergonic or exergonic? Anyone know? Good, so I have an exergonic. Everyone agree? All right, so it is exergonic. So is this spontaneous or not? Anyone know if this is spontaneous or not? So yes, so key thing with this kind of question is remember that exergonic reactions are spontaneous. They will happen. So if you have a negative delta G, or if you have an equation that says energy at the end, that tells you it's exergonic. So if it releases energy, that is a spontaneous reaction. All right, so down at the bottom, we learned about enzymes. So the reason why I have this here is just to remind you to tell you that you need to know the parts of an enzyme. So the active site, the allosterase site, that's where the inhibitors bind to, just the basic how enzymes work. So you need to know how the substrates go into the active site. The enzyme pushes them together, so then they form the product. I'm just going to make sure that you know that. Right, also in this chapter, we talked a little bit about ATP. So we talked quite a bit about ATP. So you should know the basic structure of ATP. We talked about how ATP is made by adding a phosphate group to ADP. So you phosphorylate ADP. And so in order to do that, you need energy. So that's an endergonic reaction. When ATP is formed, then it will break off the phosphate group. And that's an exergonic reaction because it releases energy from doing that. And so this ATP being making and breaking down is what runs the functions of the cell. So that's one thing. It's definitely one thing we learned about. Another thing is we learned about how enzymes work. So don't forget that we learned that enzymes, what they do is they lower the energy needed to start the reaction. Excuse me. And that's called the activation energy. So this is what happens in a reaction when you don't have an enzyme. So you still need some energy to start it, even if you have an exergonic reaction like the one there. What an enzyme does is it lowers that, so it doesn't take as much energy to cause reaction to happen. And the reason why is because it forces chemicals together, so you don't need the energy. So remember, the energy is needed to get the molecules to move quicker so they bump together. So you don't need that if they're already forced together by the enzyme. All right, so some other things we learned about with this chapter is we learned a little bit about the inhibition, and we also had some charts that we also did similar ones in lab. So looking at this, what I have there. All right, let's do the inhibition piece first. So pictured in the left, that is what a normal enzyme and the normal substrate look like. In the second picture, the blue is the inhibitor. So what kind of inhibition is this? What kind of inhibition is this? Anyone remember? So I'll give you a hint. The inhibitor is competing for the active site with the substrates. All right, so that one is called competitive, right? So it's called competitive. All right, so if that's competitive, what's the other one then? The inhibitor there is an orange. So it's binding someplace else other than the active site. So that one is non-competitive because it's not competing, right? So not competing. The next thing, let's talk a little bit about if I gave you a picture and asked the question, you should be able to tell me some information about these charts or something like them. So let's just ask the question here. Okay, why are you not working? All right, let's just say that I ask you what happens to the enzyme activity of a human enzyme when it's at 60 degrees. All right, so you would take a look at that chart. You would look at the human enzyme curve and then go, okay, let's see where 60 is. Oh, well, the curve's no longer near the 60-degree point. So the enzyme doesn't have any activity here. Does anyone know what the reason is for the enzyme not having any activity at that temperature? Guesses? Anyone remember? It starts with the letter D. All right, good. So it's denatured, right? So it's denatured. All right, so what happens down in the bottom? Let's just say that at a pH of 5, how will that affect the activity of the two enzymes that are there? Let's just say that, why does it stick in? All right, let's just say that you put these two enzymes in a pH of 5. What's going to happen? Right, so they're going to be denatured here, right? So neither enzyme is going to work because at a pH of 5, it is out of range, right? So good. So it's going to be too acidic for the trypsin. For the pepsin, it's more basic, right? So it's going to be more basic for that one. So great answer. All right. Also, don't forget, Picture reminded me to tell you that phosphorylating things activates them. That's why when ATP adds a phosphate group to a protein or adds it, like for the sodium potassium punt, it gives it energy. Any questions for me? I want to say that's the end of that chapter. All right, so next we're going to look at chapter 7. Chapter 7. All right, so chapter 7, we were introduced to other concepts here. So we learned a little bit about this process. I don't know why I had that enzyme, that reaction there. Anyway, so this reaction here, hopefully it's familiar with everybody. So oxidation reduction reactions. So they're always paired together. So the one at the top, which I gave the answer away, is an oxidation because A is losing electrons, which means that B is doing what here? Chemical B is doing what here? All right, so it is being reduced, right? So it's being reduced, right? Don't forget, Leo the lion goes grr. So we talked about this quite a bit in this chapter. So one of the reasons why I talked about it is our electron carriers, because electron carriers took the electrons and hydrogens, and so they become reduced, and then they drop them off at the electron transport chain, and they lose electrons and hydrogens, so they become oxidized. All right, so for the exam, definitely make sure you know what ATP does, what NADH or NAD plus does, what FAD does, and NADPH. The last three, they're all electron carriers. Some of them are in cellular respiration. Some of them are in photosynthesis. So you should know that those are electron carriers, but where they're used. All right, so here we go. So this is cellular respiration here. So what kind of cellular respiration is this? One of the chemicals will give you a hint. What kind of cellular respiration is this? Anybody? All right, so this is, let's see, someone typed in, so aerobic, good. Whoops, sorry about that. All All right, so this is aerobic respiration. The reason why we know that is because there is oxygen involved in this. All right, so looking at this, anyone remember which chemical is being reduced here? So if we look at the two reactants, which one is being reduced? Very good. So it's oxygen, right? Because oxygen is gaining hydrogens here, which means that glucose is being oxidized here, right? So it's called glucose oxidation, right? So it's called glucose oxidation. All right, so for this particular chapter, so I have some questions here, some things to focus on your studying, because there is a lot in this chapter. This is probably one of the more dense chapters that we have. So what are some of the key things to take away with this chapter? So first question I have for you is, these are the pathways that are used to break down glucose into ATP. So we talked about 3 different ones. So those pathways are cellular respiration, so either aerobic cellular respiration or anaerobic cellular respiration. And the other ones that break down glucose to make ATP is fermentation. All right, so those are the pathways. Which pathways use oxygen? So that would be your aerobic cellular respiration. The other ones do not. And then in terms of steps, I have this diagram here because I think it does a pretty good job kind of summarizing everything that we talked about. So you need to know the steps, kind of like the Kahoot question. So you need to know the order. You need to know where it happens. So it makes it really easy. So glycolysis occurs in the cytoplasm. And the next 2 steps happen in the matrix. And then the only one that occurs in the membrane is the electron transport chain. So just follow the steps. This diagram really helps with that. You need to have an idea of how much ATP is made total because of time, and I don't want you stressing out about it. You should know how much total NADH is made, how much total FADH2 is made, and how much CO2 is made in this process. And then you should know kind of where stuff is being produced. For example, pyruvate oxidation, there's no ATP produced there. All right, so that's a great question. So the total that is produced, so we're going to go with, I want to say the textbook said 32. With that, I'll make it quite obvious which ones it's not for that type of question. I'm trying to remember off the top of my head which number, because there's always different numbers for different textbooks. So I will let you know once I start writing some more questions for that. All right, so good question on that. Let me see what else we have. Again, make sure you know the role of NADH and FADH2 and the electrons and the hydrogens during the electron transport chain. So they all have a role. We also talked about 3 different ways that ATP is produced in the cell. So we talked about substrate level phosphorylation, we talked about oxidative phosphorylation, and we talked about photophosphorylation. So you should know those 3. All right, so then I have the steps broken down for you so you can see them. All right, so the first one is the energy investment phase of glycolysis. That's the first step. So this step is you need ATP because you need to restructure glucose to yank the energy out of it. And then the second part is called the energy harvesting phase, because that's where we make the ATP and that's where we get the NADH produced from yanking the energy out of splitting of glucose. So don't forget, that's all glycolysis is splitting of glucose in half. All right, so the diagram that's to the right, that's pyruvate. That's the products at the end of some of the products at the end of glycolysis. So notice that's in the cytoplasm. So glycolysis occurs in the cytoplasm. So pyruvate is going to then enter the matrix where it's going to be oxidated. So that's the next step. So it's oxidated because you remove a CO2 from it. And then as a result, electrons and hydrogens are added to NADP or NAD plus to form NADH here. So no ATP. And then coenzyme A is added to the acetyl group that is left over to form aceto-CoA. Right, so that's what happens at the end of that process. And so then aceto-CoA is the molecule that then enters the Krebs cycle or the TCA cycle. And so that's where you complete oxidation for glucose. That's where you break it down into individual CO2s, and you produce a whole bunch of NADH, FADH2, and then you also make ATP here as well. All right, so let's take a look at this really quickly because I know it's been a while since we've looked at this one. All right, so first one is, what is the role of hydrogen ions? So remember that the electron carriers, so you have NADH and FADH2, their job is to drop off the hydrogen ions. And then the hydrogen ions, their job is to provide energy to ATP synthase to make the ATP here. Remember, they're going to come through that enzyme, use it as a channel because it's found in a high concentration outside the cell. And so it will want to come through where there's a low concentration. And so when it does, it provides energy to make ATP. That is the proton motive force. Electrons, what the electrons do. So the electrons, what they do is they help to shuttle hydrogen ions across the membrane. That is their job. NADH and FADH2, they're the electron carriers, so their job is to drop off the electrons and hydrogens to the chain. So they collect them through all the different processes that we talked about. And then oxygen gas. So oxygen gas is important is because they collect all the hydrogen ions and the electrons, so you can keep the gradient going, and as a result, it forms water. All right, so does anyone remember the method? That is, what type of transport is used to get the hydrogen ions back inside the matrix? Does anyone remember what kind of transport that is? We'll definitely need to know this one. All right, so it is passive. Does anyone remember what kind of passive transport it is? Starts with an F. All right, good. So it's facilitated, right? Because it's a protein. It's the only one that's passive that uses a protein. All right, so the enzyme channel used to make this happen, that is ATP synthase. All right, so there's just some key things that you really need to focus on in this chapter for this part. Now I'm going to keep on going until I get questions if you have any for me. All right, last part of the chapter, we talked about fermentation. That's what's on the left. So we talked about lactic acid fermentation and alcohol fermentation. Again, this process is important because it helps to make ATP when there's no oxygen. Some ATP is better than none. And then the other job it does is it recycles the NAD plus back to glycolysis so you can make some more ATP. So again, so you want to make sure you know the two types. And what they're used for. All right, so we talked about that. The three ways of making ATP are right there. So I have those pictured right there. So make sure you know the three ways of how it's done. So again, that's probably going to be a definition type thing. All right, so I put this chart in here because this is part of the aerobic respiration extra credit sheet that we didn't really get to go through. So I figured I would go through this with you. As a result, since we've been on here quite a bit and we still have another chapter to go, so when we just go ahead, I have the answers, so you can go ahead and check this later to see if you have the correct answers or not. Again, please don't memorize this chart. There's not going to be a chart on the exam. This is purely more based on how you kind of remember each process. So take each part and know that part. So go through glycolysis. Don't go to the next step until you understand what's happening in glycolysis. Then move to the next one. And then draw this. Drawing it helps a lot. Or saying it out loud. So if you're more of an auditory learner, say it out loud. If you're more of a visual or hands-on, you may want to draw it out. So anything that kind of helps you to walk through it a couple of times. Have a chart like this or have the lecture notes handy to help you walk you through it. And so I have this chart here too, because it has the same kind of information in it. So make sure that you know this. Again, I'm not going to give you a picture and you need to fill it out again, because we don't have time for these questions. It's definitely going to be more, some application, not as much as I would like, but there'll be some application and a lot of questions. All right, so the last chapter, let's finish this up and then I will field any questions. So the next step we're looking at is photosynthesis. So we talked about different categories of how organisms make their carbon sources. Definitely make sure you know those definitions, because we talked about autotroph, heterotroph, photoheterotroph. We talked about phototroph, chemotroph, so make sure you know those definitions. We talked about the parts of the plant. Make sure you know the xylem and the phloem. We talked about parts of the chloroplast, so the granum, the thylakoid, the stroma. We talked about all those, so make sure you know those. We talked about the guard cells that surround the stomata, so we talked about them too. So when they're turgid, the pores are open, or the guard cells are open. When they're flaccid, meaning the water has left because it's hot, it will close and shut those pores so the water doesn't evaporate. But also keep in mind that the gases can no longer exchange either because it's shut. We talked about visible light spectrum being the range that plants use. That's the type of radiation they use. So looking at this picture here, which colors of light are the best for photosynthesis? Which colors of light? Anybody? Colors of light. All right, so good. So the violets and blues and the reds, right? So those are the best. Which ones are the worst? Which ones are the worst? Good, so green, right? So green and yellows. Easy way to remember it, leaves, you see them, they're green. Why? Because the plant doesn't use them. Right, so it's the easiest way to remember that. Those are your 4 pigments that we talked about. We talked about them in lab. Why do we have multiple pigments? Why do we have multiple pigments? All right, so refresh everyone's memory. We have multiple pigments to broaden that range, right, that you can absorb, right? So chlorophyll, which is chlorophyll A, which is the main pigment, only absorbs at a certain range. If you look at the chart, that's the teal looking color. But in order to absorb more, you need your other pigments. Also, don't forget that the carotenoids also do photoprotection. So they help protect chlorophyll because it is light sensitive. It doesn't want to absorb too much light or it actually breaks down. All right, so some of the things we haven't really talked, so one section I haven't really talked about these parts yet, which is light and dark reactions. So you definitely want to make sure you know what happens in the light reactions. That is where light is absorbed and it's converted into ATP and NADPH, which is the chemical energy. As a result, water is split because electrons are needed to replenish chlorophyll A. And then as a result, you form oxygen. So this is where oxygen is produced. That ATP and NADPH are going to provide energy to run the Calvin cycle, because the Calvin cycle is where you take CO2, you add them together to make sugar in the form of glucose. And then the components of that, so the ADP and the phosphate and NADP plus go back to the light reactions to collect more electrons or provide more energy so you can keep the cycle going. All right, so don't forget that the light reactions occurred in the thylakoid membranes. The Calvin cycle occurs in the stroma. I already asked that question. I forgot it was on this slide. All right, so this look at an experiment. Because of the time on the exam, we won't be able to do more questions like this, but I want to make sure we kind of do some application questions here to see if everyone's on point. All right, so say you put a plant in green light and you wanted to see how it affects growth rate, which is in mass over time. Let's see if you remember, what is the independent variable here? What is the independent variable here? It's a good final question. Good, it's the light, right? So the light is the independent variable, which means that the dependent variable is the, what is the, good, so growth rate. All right, so control variables, we have quite a few here. So remember, those are things that are going to be the same between the two. So we have a lot of different ones here. So we have the plant, correct? We have the soil, that's a good one. Water, air, any of that stuff, all that stuff's going to be the same. So which one is the control setup? The one on the left or the one on the right? Which one is the control setup? Remember, the control is the one that's the side that normally happens. Yep, so good. That's the one on the right, right? Because you usually just have regular white light on the plant. So that means the experimental one would be the green one, right? Because that's the one that's not typically done. Okay, so keep those in mind, definitely for the future, for the final. All right, so some of us haven't had this yet, so I apologize for them because we're going to talk about this on Tuesday. So this is the light reactions here. So some basic takeaway things here, because you're not going to draw this, you're not going to do any of this stuff, you're not going to have a diagram or anything. Some really key takeaways. The first thing is that these photosystems collect the light because the pigments, those photosynthetic pigments are in these photosystems. So that's the purple pieces there. So that will, the light will hit those. It's going to excite these pigments and pass energy to the chlorophyll A's in the middle. That's what those P's are. So those are actually special chlorophyll A's. You do need to know that photosystem 2 has P680. Photosystem 1 has P700. That's a special chlorophyll. Photosystem 2 is the first one in the system. It is the one that water is split and gives electrons to P680 so it can continue passing electrons on. As a result, oxygen is produced. So oxygen is produced in photosystem 2. The electrons from photosystem 2 travel down to photosystem 1 to replenish the electrons for P700. So photosystem 1 also collects its own energy as well. So P700 in that photosystem will pass electrons down a chain, which will then eventually go to make NADPH. All right, so the ATP that's made during photosynthesis is made when it travels through carriers between photosystem 2 and photosystem 1. So this is called photophosphorylation. Very similar concept as the electron transport chain in cellular respiration. So it's using chemiosmosis and then making ATP using oxidative phosphorylation. Similar steps. All right, so this is kind of a blown up view so you can kind of see that whole process. So in the middle, that is the light reactions. If you notice in the middle, hydrogen ions are being pumped in. They're also being introduced when water is split. All right, and so those hydrogen ions are important because they're going to build up inside the thylakoid. And so that's going to provide a gradient for ATP synthase again. So then the hydrogens are going to travel through ATP synthase and then make ATP, which will then be used for the Calvin cycle or to run things in the plant. NADPH is produced when the electrons come from photosystem 1, and then they get added to NADP+. Right, so both of those are needed to run the Calvin cycle. Because remember, if you have no light reactions, you make no ATP and you make no NADPH, so that means you have no Calvin cycle, which means you have no glucose. So light reactions are important. Okay, so this is kind of a brief overview of the Calvin cycle. So there are 3 steps to the Calvin cycle. So you have carbon fixation, reduction, and regeneration of the CO2 receptor, which is called ribulose bisphosphate, or RUBP. The enzyme that adds CO2 to RUBP is called Ribisco. I remember Nabisco. I remember that since middle school, so that's how I remember it. But that's how you introduce the CO2s into the cycle. ATP and NADPH are used in the carbon fixation piece to add energy to it. And then during the reduction piece, this is where you have G3P being produced, which is glyceraldehyde 3-phosphate. This may sound familiar to some of you because this is produced during glycolysis too. What happens is a couple of these are added together. So you actually need the cycle to go through 6 times in order to make 1 glucose in this process, because you only yank one of these G3Ps out every time the cycle goes through. You actually produce 5, you produce 6, but only one of them gets to be used to make the sugar. So it actually takes a while to make it. So what will happen is most of the G3P will go to remake ribulose bisphosphate. And so that's the last part of the process. You need ATP to make that. So this kind of has everything about the Calvin cycle right here. So please make sure that you're familiar with these. Hopefully the homeworks as well as the Quizlet will help with that. All right, and this is the last slide here. This just kind of summarizes all the parts, what's happening here. So sugar, if there's a lot of it, will be stored as starch. And then when it travels through the plant, usually glucose is stored as sucrose, and then it travels throughout the plant. Using phloem, right? The phloem one, right? So don't forget about that. I want to say that's my last slide. All right, so does anyone have any questions for me in terms of concepts, anything like that? Once the exam's written, I can give you more ideas of what that's going to be once that's done, but that's the next thing that has to get done. Once I get all the classes need to be set up first. That's the key piece. Does anyone have any questions for me? I keep checking to make sure anyone's here because everyone's so quiet. Any questions for me? So this is recorded. So what will happen is once we're done the meeting, I will submit this so it can be translated, and then hopefully by tomorrow I will post it. Hopefully, we'll see how long it takes. But I definitely would review this again. Hopefully it's helpful to you. And again, you can always ask me questions as well because you know I'm home. I'm not going anywhere. So if you want to ask me questions online, you can always ask me that. All right, so does anyone have any questions for me before we log out of here for tonight? Correct. So it is on, the exam is not until the 8th or the 9th, right? Because we don't start back out until then. So you have plenty of time to review this. All right, does anyone have any questions for me? All right, so this is the way we're going to go for lectures. So hopefully this was okay for everybody. This is what we're going to be using. Again, if you have any questions, you can always email me. Please think positively. You can do this because now you can watch this again and review it. Have faith in yourself. Again, if you start feeling alone or stressed or anything like that, please feel free to contact me at any time. And And any last minute questions, because I'll log us out. All right, thanks so much for showing up. I really appreciate you coming. And I will see Monday, Wednesday, Friday on April 6th. That's when we start back up. And Tuesday, Thursday, I'll see you on April 7th. All right, look for my email with the link on it. All right, I will talk to you in about a week. Have a great night, everybody.